Hello everyone, my name is Laura Modelli and welcome to Science for Education, supporting teaching and learning in science education. In today's episode, we are going to do a dissection activity with a sheep eyeball. And before we start, I would like to say thank you very much to our friend Neil for his support by sending this eyeball specimen. Thank you, Neil, for helping us to make this episode possible and contributing to our educational science videos. To do this activity, we are going to need some materials such as forceps, teasing needles, scissors, a scalpel, a dissection tray, and a sheep eyeball. We will also need to put on our safety equipment like safety goggles, gloves, and lab apron or lab coat. I have created an activity sheet that you can use to follow along with this video. You can find the link to this activity sheet in the description below. As we are doing the dissection, we can use the eyeball diagram to help us know which part of the eyeball that we are going to observe in this activity. And perhaps you are wondering why we chose a sheep eyeball for the dissection. A sheep is a mammal. Therefore, some organs of the sheep share the same structure as ours, humans. So, by studying the sheep eyeball, we will not only learn about the sheep organ structure, but also we will be able to learn and make comparison to other mammals and humans. Let's get our hands dirty. We are going to start with observing the surface of the eyeball. On the surface here, we have something called the cornea. You can see our specimen is a little misshapen from the packaging. Here, the cornea looks cloudy as a side effect of being preserved, but in a living animal, the cornea looks clear and transparent. The transparency of the cornea allows the light to enter the eye. Next, we have the sclera. The sclera is also the outer part of the eyeball. And as you can see here, the color is opaque and feels a little tough. It is around the eyeball, but not at the front. The sclera protects the inner structure of the eyeball and maintains the rigidity as well. The next part is the optic nerve, but before that, we can trim the excess of the sclera, to kind of clean the eyeball up a little bit. This is just some excess fat and muscles attached to the sclera. You will find this optic nerve in the back of the eyeball. The optic nerve consists of a bundle of nerve cells that will send signals from the eye to the brain. What does that mean? It means when the eyeball sees things, the optic nerve will send the information to the brain, and the brain will decide what is there. Now we are moving to the inner part of the eyeball. We're going to cut the eyeball in half. We will hold it gently on the pan, and my thumb and forefinger now are holding the cornea and the optic nerves. We're holding and cutting in this way because we don't want to cut the lens. We want to save it for our observation. We will make a small incision over here with our scalpel, and then we are going to use our scissors to continue cutting. Again, we are only cutting the sclera. We don't want to squeeze the eyeball because we want to save the interior parts. Here, you might see that there is a gelatinous transparent material starting to come out. This is called the vitreous humor. And as you can see, there is a cavity between the retina and the back of the lens. This cavity area is called the vitreous body. And the vitreous humor fills this cavity and helps maintain the shape of the eyeball. Now, we are going to observe more of the inside part. Let's refer to the diagram again. What are we looking for here? So we are looking at the inside and front part of the eyeball. Here we have the lens. This lens is hard and pink as a result of being preserved. This lens is a biconvex transparent structure. The function is to focus the light coming into the eye through the cornea and pupil. Here we have hyaloid fossa, so it is an indentation in the center of the vitreous body. So the hyaloid fossa supports the lens. The surrounding area of the hyaloid fossa is something that we call zonula ciliaris, made up of suspensory ligaments that suspend the lens and stretch it to focus. Next, we are going to turn this front half of the eyeball over, and we are now looking at the cornea again. We are going to cut the round side of the cornea to remove it and then cut it in half so we can observe the thickness of the cornea. The thickness of the cornea changes over time. The older you get, the thinner your cornea will be. Next, let's find the iris. Using the forceps, we are going to carefully separate the edge of the iris from the inner surface of the eye. The iris is a diaphragm that regulates the size of the pupil. So, when there is light coming into the eye through the cornea, the pupil will change size to control how much of the light can come in. It's easy to see how pupils work in cats. In humans, remember, when you are coming out suddenly from a dark room out into the sunlight, your sight turns black for a moment. That is because your pupil takes a little time to regulate. In humans, different people will have different iris color: blue, green, gray, hazel, brown, and black. Here, as you can see, there are dark lines around the hyaloid fossa. These lines are pigment from the iris. Next, let's do some more observation on the other half, the back part of the eye. Here we have the retina. The retina is the light-sensitive portion of the eye composed of receptor cells called cones and rods. Cones are responsible for perceiving color, and rods are responsible for perceiving difference in light intensity from dark to bright. Now, if we follow the mass of nerve cells to the back of the eye, just right where the optic nerve begins, we will find the blind spot. Then if we turn this back and pinch a little over here, we might be able to see the fibers of the nerve in the optic nerve. These fibers carry all the information to the frontal cortex in the brain. Let's go back to the inside part of the back. If I move the retina here, we'll see the choroid, the thin dark layer between the retina and the sclera. And this portion of the choroid, you can see it appears to be iridescent blue and green with shade of yellow. It is called the tapetum lucidum. This part helps in night vision by reflecting available light back onto the retina. Humans don't have this tapetum lucidum, only animals have it, and that's why their eyes will reflect the light back. This is very clear when you see them in the dark room or at night. Now that we have learned parts of an eyeball and their functions, before I end this video, I would like to summarize how the light passes through the eyeball in order. So everything that we just learned will make more sense. Light enters the eye through the cornea. From the cornea, the light passes through the pupil. And from there, it then hits the lens. Next, light passes through the vitreous humor. and then the light reaches the retina. There, the image is inverted. Then finally, the optic nerve sends the signal to the visual cortex of the brain. The visual cortex turns the signal into images, which is what we perceive as our vision. I hope that you have had the best time learning about eyeballs, all the parts, inside and out, and all of their functions. I will see you next time. Bye.